Thank you very much, Kenny. So my name's Dr. Chris Hatcher. I'm a lecturer in Practical Ecology at the University of Gloucestershire, and I'm going to be talking about a paper I recently published in Functional Ecology entitled Carnivorous Sundews Are More Carnivorous in High Light Bog Microhabitats that Are Not Also Nutrient Rich. So the luxury of presenting to a group such as yourselves is that I can start the floor just talking about carnivorous plants, which is always nice. And plant carnivory has evolved independently numerous times, at least 10 times. And we see from the figure here, there have been some recent additions in and around this area with Triantha, which is an interesting one that's carnivorous just on its flower stems. And there's also been some proto-carnivorous plants in this sort of area of the taxonomy that have carnivorous seeds. The area that I'm talking about is with Drosera, that they have the same carnivorous ancestor as this purple block here, the Nepenthes. So closely related to Venus flytrap and its aquatic snaptrap sister of the water wheels there. But carnivory's evolved numerous times. A common misconception is that they're carnivorous to gain energy from their prey. That's not why they're carnivorous. It's to supplement low root nutrient uptake. Carnivorous plants photosynthesize like other green plants. As far as we know, they're all C3 photosynthesizers. And benefits of carnivory are quite numerous as well. So they can increase growth rate, they can have earlier flowering, increased seed set, it can increase the rate of photosynthesis. Interestingly, it can increase root activity, and it can also increase seedling growth as well. But there are also associated costs with being carnivorous. There's a twofold cost from the production of traps on leaves because it requires a resource just to create the traps. But by switching to that strategy, there's also a reduction in the photosynthetic output as well. As well as other things like for the sundews that produce mucilage, that's carbon-based. Traps tend to have extra floral nectaries, so they'll produce nectar. They might produce scents to attract prey, these volatile organic compounds, that's just those scents. Or they produce enzymes, and they have to actually assimilate the nutrients. They have to take up the nutrients somehow. So there's lots of costs with being carnivorous as well. And that means that there's a trade-off between the benefits they get from carnivory and the costs associated with that strategy. And Tom Givnish in 1984 published a cost-benefit model for the evolution of carnivory. And this model demonstrates that carnivory is evolutionarily favorable when there are marginal benefits as a result of allocation to carnivory. And these two figures here just highlight where gains can be made under certain environmental conditions. So what we see here is this blue dotted line is that net gain in terms of carbon associated with allocation to carnivory. And where you get the dotted line going up, that means any increase in how carnivorous they are will provide an overall benefit, those marginal gains. And you see that in sunny, moist conditions compared to those shady or dry ones, and also in these infertile environments here where you get this uptick in curve as well. So this model was originally put forward to explain the evolution of plant carnivory, but provided that investment in carnivory is a plastic trait, it's something that's variable, they can change how carnivorous they are, then you can consider this evolutionary model in an ecological context. And what that means is that across variation, either within a site or across sites, you would expect the plants to also change in terms of how carnivorous they are. And that can be measured either in the carnivorous traits that they express, or it can be looked at in terms of the proportion of nutrients that they get from their prey compared to through their roots. And what this means is that they are restricted to these kinds of habitats where there's low soil nutrient availability, high water availability, and high light intensity. and those are generally peatlands or bogs. There's lots of nomenclature around what kind of wetland you call them, but generally they look a little something like this. I say usually here because there are a few exceptions. I think it's Triphyophyllum that lives in a very dry habitat, but it's got a taproot that goes down. So these are the general rules for carnivorous plants. And there's been some research on to what extent carnivorous plant traits are an adaptive trait. And what we see is that generally they follow the predictions of that cost benefit model when applied in an ecological context. So to take a look at these examples, Drosera rotundifolia, which is that round-leaved sundew that we're looking at, that's in these images that we see here. We find that they can increase trap stickiness, so the mucilage that they produce in low nutrient, high light conditions. We see that in areas where there's increase in nitrogen availability through the roots as a consequence of atmospheric N deposition, then we see a decrease in the proportion of nutrients derived from prey. That's this figure here. Shows an amount of N derived from prey goes down as you see an increase in this atmospheric nitrogen deposition. And we see this across various different species of carnivorous plants. So Sarracenia do a similar sort of thing to experimental N addition. They can change picture morphology in response to light availability. And Pinguicula, those butterworts, they're another flypaper carnivorous plant. They can increase that mucilage secretion or gland density in response to changes in light availability as well. So what we see here is that the importance of altering investment in carnivory as a mechanism for adaptation to different habitat conditions. What we predicted within this paper is that those same sort of patterns that we see in the literature are likely to occur in response to very small scale habitat variability as well. So studies of variability in carnivorous plant traits have generally considered broad scale continental differences among sites like that when looking at nitrogen deposition, or they're manipulative studies or consider within site changes over time. I know Steve Brewer's done a lot of work on that with Sarracenia, but there was scope for understanding how these responses translate into patterns of carnivorous plant traits at very small spatial scales. And this is particularly important if these small spatial differences exist in their natural habitat. And within pattern peatlands, in which you find carnivorous plants, small scale patterns of habitat variability tend to persist for quite long periods. So this is an example of a patterned peatland in Sweden. This is an orthomosaic. So we've flown a drone across the site and then stitched a lot of photos together to get this very high definition image of the bog as a whole. And what we see here is this microtopography where we have hummocks, these raised areas, which are the sort of darker green bits on this image, and hollows, which are these sort of paley purple because of the kind of imagery we've used here for these lower, wetter sites, wetter areas of the site. And those differences persist for quite a long time, which means that there's sufficient time for phenotypic or genetic adaptation to these environmental differences, particularly when compared to those sort of short-term studies that have been conducted. So these patterned peatlands, like these, are characterized by these microforms or microtopography that you see here, these hummocks and hollows, these higher and lower areas. So those raised hummocks, you might know those as strings, and then these lower hollows and lawns, you might know those as flarks, depending on where you are in the world and what kind of terms you use for this. Because of how these peatlands organize into these microform patterns, these habitats are extremely variable at small scales. So this is the peatland in Sweden. This is the same peatland, and you can see this variation. This is a researcher in the wild doing some work here. We've got these sort of darker areas here are these hummocks that are raised, and then they're characterized by these lawns and lower, wetter areas that are the hummocks that you can sort of see. And as the hydrology changes, you end up getting sort of orchids and horsetails going into these trees in the backdrop here as well. And the pattern of differences in these hummocks and hollows will vary because of the processes involved in what's forming that microtopography and how the nutrients are cycling within these areas. And that changes depending on the local climate conditions. So the formation of these hummocks and hollows is due to the rate of peat accumulation in areas at the site. And this peat accumulation is driven by evapotranspiration and precipitation. So essentially, a ratio of how much rainfall there is compared to how much of that water is drawn back out of it in processes of evapotranspiration. And there was some work conducted in 2010 by Martin Epinga that found that peat accumulation within these sites was driven by that pattern of precipitation evapotranspiration ratio. And then once you have these characteristic hummocks and hollows, then that ratio of precipitation evapotranspiration determines where nutrients flow within these sites. So in areas where you have a very high ratio of precipitation, where there's lots of rainfall, then the nitrogen within the site goes down into these lower areas in these hollows. Where you have a much lower amount of precipitation compared to evapotranspiration, then the nutrients get drawn up into those higher areas. But you also get very clear differences in other conditions within these microhabitats. So the higher areas, these hummocks, they are characterized by much more shrub canopy vegetation, which means that below that canopy, they're much more shaded in these areas. They are to an extent drier in these areas as well. And then these hollows are much more open, they're less shaded, and typically there are these wetter areas as well. This pattern of light availability, essentially, the amount of shading between hummocks and hollows, that's fixed across sites, that's consistent. But the amount of root available nitrogen in these sites changes based on that level of precipitation and evapotranspiration. So where the nutrients accumulate, either in the higher or the lower areas, is determined essentially by how much rainfall there is compared to that evapotranspiration going on at the site. Within this study, we used Drosera rotundifolia as a study system. The reason for that being that the round-leaved sundew is very cosmopolitan. You find it on every continent except Antarctica. You also find it within sites on those hummocks and in the hollows, which means that you've got the same species within both of those microhabitats. They're relatively easy to study. We have broad understanding of their responses to nitrogen availability, and we, based on studies of other sundews, have a good idea of what their response to changes in the light environment are as well. And the other thing you can do with carnivorous plants is measure investment in carnivory through two different sort of strategies. The first being looking at carnivorous traits. So some studies look at the stickiness of the mucilage. You can look at the density of the hairs, these trichomes or tentacles as they're referred to. And you can look at the trap area for capturing prey. So you can look at plant traits for investment in carnivory, or you can look at the proportion of nutrients derived from prey. And that's through using nitrogen isotope analysis, because as you go up trophic levels, they take on more delta 15N, which means that you can determine the amount of nutrition derived from prey compared to via the roots by doing some nitrogen isotope analysis. So the overall aim of this study was to determine how precipitation, evapotranspiration driven small scale resource variability impacts plant nutrition and variability in investment in carnivory, and linking this to how that trait variability changes with local climate. And the predictions of that were that at small scales, light and N availability will alter Drosera rotundifolia investment in prey capture. Plants growing in higher light and lower N habitats will increase investment in prey capture and reliance on prey-derived N. And this all centers back to that cost-benefit model. So we tested hypotheses. And just so we look back at here again. So this is that figure that we looked at before, looking at the microhabitats and how nutrients accumulate in different areas based on the levels of precipitation and evapotranspiration ratio we have. So A here is where predictions for which there's much more precipitation compared to evapotranspiration. And so what happens is that nutrients are drawn into those lower hollow areas. So for higher PET sites, nitrogen accumulates in hollows according to that directional flow of water. But the hummocks are much more shaded than hollows. And as a result, what you get is investment in prey capture and prey N uptake by Drosera rotundifolia at these sites being the same, whether they are found in the hollows or the hummocks. And that's because of the relatively greater benefit of carnivory in those low root N microhabitats. But that's contrasted by the elevated cost of carnivory in those low light microhabitats. So what you see is a very similar outcome because of those differences in the impact of reduced light in the higher areas, but increased N availability in the lower areas. So it sort of cancels out the benefits that can be made in terms of carnivory in those two different microsites. And then when we see precipitation, evapotranspiration ratio decreasing, where you might have something more similar in terms of that contrast, the distribution of N changes within the sites. You might have a much more balanced amount of nutrition in the higher and lower areas. And this will result in greater investment in prey capture and prey N uptake for the sundews in those lower N, less shaded hollows. So where precipitation isn't so high compared to the amount of evapotranspiration precipitation that's going on, then there's much more benefit to be made in those open, less shaded areas because the relative cost of carnivory goes down, essentially. So to do this study, we selected 3 sites that are along this gradient of precipitation evapotranspiration ratio. This map here, those dark green patches here, this essentially highlights where peatlands are. So in the UK, for example, they tend to be found more in Scotland and Ireland areas. There are some examples further south in England. They tend to be much more degraded in these areas because lots of peat extraction. Irish peat famous for peat extraction as well. But the sites we used were up in Scotland, Sweden, and Finland, and they go along this contrasting ratio in the amount of rainfall to evaporation there is at the site. And I've included N deposition in here as well. They're sufficiently low that they're not sort of saturated with root N, that you'd be able to see differences in carnivorous trait investment. And the pattern that we should see here is that in the Scotland site, we'll start at the bottom here, where you have a very high level of rainfall, then you expect the nutrients to accumulate in those lower areas in the hollows. And so you would expect their investment in carnivory to be very similar in hummocks and hollows because you've got that that contrast in terms of the amount of shade increasing the relative cost of carnivory in the higher areas, but in the lower areas you've got increased available root N, which decreases the benefit you get from carnivory. So they sort of cancel each other out, and you'll see a similar sort of investment in carnivory. And then in contrast, in the Finland site, which has this lowest level of PET contrast, you get nutrients beginning to accumulate in those hummock areas. And so the benefit of investment in carnivory in those hollows where there's lots of light availability and very little root N availability, you expect to be much higher. We looked at 25 hummocks and 25 hollows per site, with 5 plots in each hummock and 5 in each hollow. And we measured 5 plants in each plot to sort of average out the variability within those sections as well. And for measurements, we focused on 2 habitat characteristics that I've mentioned. We looked at light from above ground shade and nitrogen availability. So we measured light interception within each plot in each hummock and hollow at the sites to give vegetation coverage, phytovolume, and light transmission. So you can use a light photometer, measure the amount of light above those shrub canopies, which tends to be heather, and below, and then you can look at the percentage of light that's intercepted, essentially. And we also measured the amount of N available to the plants by measuring tissue nitrogen concentration in the sphagnum present. They're used as a bioindicator of N availability at these sites. These pattern peatlands, most of their N comes from atmospheric N deposition, and sphagnum are a reliable indicator of the amount of N available at the site, provided N deposition isn't too high, which it wasn't at these sites. So those are the traits we looked at in terms of habitat characteristics. And then for the sundews, we measured the tentacle number, or the trichome, so it's that density of hairs here, as well as the trap area as an indicator of investment in carnivory in terms of the carnivorous traits. It wasn't possible for us to look at stickiness here because we're looking at 3 different sites. If it happens to be heavily raining at the times that we go to these sites, the mucilage washes off. And so it's not a good way to test across sites, across time. But we used trichome density and trap area as an indicator of investment in carnivory. And the other thing that we wanted to do was look at the diet of these carnivorous plants. So does that investment in carnivory translate into an increase in the proportion of nutrients derived from prey? And to do that, I mentioned that approach of nitrogen isotope analysis. If you want to calculate that, then you need to look at the nitrogen isotope of non-carnivorous plants that will give you a baseline of the nitrogen isotope of a carnivorous plant if that carnivorous plant captured no prey, if it had 0 nutrients derived from prey. Then you need to look at the nitrogen isotope of prey at the sites, and that will give you a reference for a carnivorous plant that gets all of its nutrients derived from prey. And then you can take tissue analysis of the actual sundews, the carnivorous plants, and based on that nitrogen isotope ratio, you can then work out the proportion of nutrients derived from prey compared to whether they're root derived. So you have a physical idea of investment in carnivory by measuring these traits, and then you have how that's realized in terms of the nutrients that they get by doing this nitrogen isotope strategy. In terms of statistical analysis, we presented our data in 2 different ways. We did a nested ANOVA with planned comparisons for microforms within each site, and that's analyzing the raw data that we collect within each site, but it means it's not appropriate to make meaningful comparisons within those microhabitats across sites. And so to do that, we looked at a trait contrast to look among sites. And essentially what this does is looks at the difference within sites between the hummocks and hollows and creates a value from -1 to 1 in terms of the difference in those traits in hummocks and hollows, and then they are comparative across sites. And we used a bootstrapping approach to look at pairs of hummocks and hollows within each site in order to do that analysis. And the findings that we have. So to begin with, we will look at the habitat characteristics. So vegetation coverage, and we've got this this y-axis here, which is our trait contrast. And if the traits are very high above the line, then these are values for hummock habitat. And then if they're very much below the line, then it's trait contrasts for the hollow habitat. So what figure A means here in terms of vegetation coverage is that there is much greater vegetation coverage in the hummocks compared to the hollows, and that is the same at each site. So we've got Scotland, Sweden, and Finland in these figures. There's much more vegetation in the hummocks compared to the hollows, and that is the same across each site. That was a consistent pattern. And this is that trait contrast. These are those raw values in terms of those differences. So we've got the mean value with 95% confidence intervals, and then we've got the plot values in these circles that you see here. And these are statistical significance in the letters, and asterisks are also statistical significance as well. So, yeah, very clearly much more shaded hummocks than hollows. A similar pattern for phytovolume. There's much more vegetation in the hummocks compared to the hollows. And as you would expect for light transmission, it confirms that the hollows are much lighter. That's where we see these minus values. So it's much lighter in the hollows compared to the hummocks, and that is consistent across all sites. And then looking at nutrient availability, that nitrogen accumulation, this is where we expect there to be differences among sites because it's along that ratio of evapotranspiration precipitation. And what we see that in Scotland, where there's much greater precipitation, there's a lot more rainfall compared to the evaporation. We expected there to be a nutrient accumulation within the hollows, and we expect that pattern to decrease across the other two sites. And what we find is exactly that. So we've got these negative numbers. There's greater sphagnum N concentration in the hollows compared to the hummocks in Scotland, which means that our predictions in terms of that ratio are apparent because the sphagnum as that indicator of N availability is reflecting that pattern. And we see that pattern diminishing as we get to Finland, where there is very little difference in terms of the sphagnum N concentration within those sites. So we've established that there are those clear differences within the microtopography. The hummocks, much more shaded than hollows, but N availability changes based on that evapotranspiration precipitation ratio. And then what we see with the carnivorous plants is that tentacle density in terms of investment in carnivory, we see much greater investment in those sites where there's a more even level of nutrient accumulation within the sites. For Sweden and Finland, we see very little difference in N derived from prey. In Scotland, we see a much greater proportion of N derived from prey in those more open areas in Sweden and Finland, where there's very little difference in root nutrient availability. So the benefits from carbon increases. And that contrast in terms of tissue N, we see that in Scotland, there's greater tissue N within the hollows, whereas there's a more even sort of pattern within Sweden and Finland. And interestingly, we'll sort of come back to this pattern later in that tissue percent N was quite high in Scotland compared to the other two sites. This is looking at that raw data that's going on there. Relating this back to our predictions. So natural occurrence of microhabitat variation. This is that figure that we looked at right at the start. And our findings confirm those hypotheses that we had. So very clearly, we saw that in the area where precipitation is much higher than evapotranspiration in that Scotland site, we see very similar investment in carnivory within the sundews in hummocks and hollows. But because that relative cost of carnivory is lower, we see a greater tissue N in those sites in Scotland compared to Sweden and Finland. And in these sites, and these are the examples of that Sweden and Finland sites, we see much more benefit can be had from the plants in the hollows in Sweden and Finland, where it's more open, the relative cost of carnivory goes down. And so they invest more in carnivory and have a greater proportion of N derived from prey. So some implications. So we confirm that yes, PET controls nutrient distribution in pattern peatlands sufficiently to drive trait variation in microhabitats. It extends our understanding of the adaptive function of carnivory to much smaller spatial scales. Ecological impact of a climate-driven variation in within habitat heterogeneity is likely to be different in different peatlands, and that's because of those local climate conditions. This video that we're looking at here is an example of a minerotrophic peatland. So it'd be interesting to see how differences here are apparent as well. So that you do see that same microtopography within minerotrophic peatlands, but that can also vary in terms of these sort of tussocky higher areas. So it'd be interesting to see whether we see those same patterns in slightly different wetlands as well. An interesting talking point is that Scotland's site had elevated tissue N levels across sites. That's likely to be because of the amount of prey availability that's there. The Scotland site was very clearly incredibly dense with midges and mosquitoes. It wasn't the most glamorous of sites, but the sheer prey availability was likely what was elevating that tissue N overall. And then some other interesting things of note here. So what are the implications of our findings? Well, we've found that a climate-driven process specific to local areas is sufficient enough to drive changes in plant traits. It'd be interesting to see how that impacts on the non-carnivorous plants as well. Trying to predict the future is a real challenge here because we've got microhabitat responses to local climate conditions, and it's quite difficult to predict how the sites are going to change over time. You know, global average temperatures are going to be increasing, but it's a bit more nuanced in terms of what's going to happen at specific sites. So wetter sites might get wetter and drier sites might get drier. What's going to happen to those that sit on that even level of evapotranspiration precipitation? What implications does that mean for the differences between hummocks and hollows? How will that impact on species that specialize to just those microhabitats as well? Something else of note is that we used Drosera because it is so widespread. It's a cosmopolitan species. You find it everywhere, essentially, in quite high densities in these peatlands. What makes them so successful in these areas? Is it because they have this very high propensity to adapt to these different conditions? Are they more plastic than others? Are they able to tolerate a greater range of conditions? Perhaps they are, they're much more generalist because they're able to adapt to those differences. There are some sites that I've been to in the UK where the shading they experience is incredibly high, upwards of 75% light interception, and they're persisting there and doing fine. So looking into that dynamic is interesting. understanding Drosera interactions with other carnivorous species that they tend to live in the same area as well as some interactions with other predators. It would be interesting to look at the responses of. I know some work's been done on spiders, toads, and Drosera as well. And then finally, just some acknowledgements here in terms of the funders and people associated with helping facilitate this research. So thank you very much for listening. Hopefully you've got some questions in terms of the implications of this research. I'm happy to take some now.
Okay, thank you, Chris, for an excellent presentation. And this was part of the ICPS's monthly webinar series. The ICPS is an organization of culturists, conservationists, scientists, and educators all interested in sharing the knowledge and news of carnivorous plants. We've been around since 1972, so over 50 years. Since then, we've expanded, and our membership and our perspective has changed a little bit. While the society was once a club for just plant growers, we have now evolved into a truly international society with a professional quarterly quality journal, and we are an increasing voice in the world of conservation and a network of horticulturists larger than ever before. Our next webinar is going to be in a couple of weeks by an author and illustrator from Australia who's going to talk about the wonderful world of carnivorous plants. The first Wednesday of May every year is World Carnivorous Plant Day. You can go to our YouTube page to see all of our past videos. So on World Carnivorous Plant Day, we get over 24 scientists, backyard growers, nursery people to talk about carnivorous plants. And you can watch all of the archived information back there. And what's exciting about this year, March 7th, 2025, it is the 5th anniversary of World Carnivorous Plant Day. You can participate. We are looking for content, and the videos for World Carnivorous Plant Day need to be submitted 1 month from today. And we're looking for the theme of 5. So if you have a video idea that has the number 5 in it, we will probably accept it. So we're looking for things like 5 easy plants to grow indoors, or 5 fertilizers you can use on carnivorous plants, or 5 fertilizers you can use on nepenthes or Venus flytraps, or 5 pet store foods you can feed carnivorous plants, or 5 carnivorous plant species you can find in Scotland, things like that. So we're looking for the theme of 5. And Chris, I added this slide when you were talking because for our first year, World Carnivorous Plant Day, we had middle school students interview lots of different carnivorous plant experts. And of course, one of the first people I reached out to was Tom Givnish, because he discovered, or he named, one of the first carnivorous bromeliads, which is cool. But what's maybe even more important is that he created the first modern definition of what is a carnivorous plant. And I think it's crazy to think that, like, just in the 1980s, we did that. And the person who said, what is a carnivorous plant is still alive today. All right. If you want to participate in World Carnivorous Plant Day, send me an email at kenny@carnivorousplants.org. All right. Thank you, Chris, for that brief commercial. And I still can't connect to my video, so we're just going to read you questions.
Okay.
Ben Benjamin from Florida says, to what degree can generic sundews absorb nutrients from their roots? You mentioned that carnivory supplements nutrient uptake. I noticed that my plants may still grow without feeding or for months, but eventually may suffer. And that kind of brings us back to the ICPS is really the nexus of scientists and growers and nursery people. So everybody who likes carnivorous plants. So Chris, does your background help Benjamin at all?
Yeah, sure. So what do generic sundews absorb nutrients from their roots? So sundews will take up nitrogen through the roots, and if you do as you say and you completely prevent them from capturing any prey through the leaves, generally they'll just grow slower. So it depends what you mean by suffer there, but generally they'll just grow much slower. Um, and that applies across the developmental stages as well. So I know that there are some growers that have a sort of a special mix to that's essentially a fertilizer medium that they're growing them in. And because they're in that greenhouse setting, they don't get out-competed like all of the other plants that they otherwise would in situ if there was that level of nitrogen deposition going on. In their natural habitat, if you get lots of N deposition and a change in the amount of root nutrients available, generally what happens is they get out-competed by other plants rather than it having a much more significant impact on them directly. So you can get them to grow faster by adding nutrients through their roots.
All right. Tim from West Virginia in the US says, can we extrapolate that other carnivorous plants would be more carnivorous in shaded or nutrient poor environments, just like your study showed sundews?
Yeah, so my study follows the cost benefit model that you mentioned, Tom Givnish published, and that was with that bromeliad as well, actually. Um, and that predicts that in areas where there's lots of light and very low root nutrient availability, then the carnivorous plants will be more carnivorous. And that's because the cost of carnivory goes down in those lighter areas and the benefit of carnivory goes up. So you would predict that other carnivorous plants, not just Drosera, would also be more carnivorous in those more open areas and those more nutrient limited from the roots. So yes, it should be applied to other plants. I suppose the other consideration there is whether their carnivorous habit is plastic, whether they can vary how carnivorous they are. Um, but I would expect to an extent, yeah, other carnivorous plants would follow a similar sort of pattern.
Swan Yi from Hong Kong, but studies in the US, says, was there any measurement done on ground level floor depth below the surface?
No, and it is one of the things to consider, and because water is obviously important for carnivorous plants. So we didn't look at where the water table was in our sites, but our sites were all very wet, and hummocks and hollows were sufficiently wet for those plants to occupy those areas. So yes, to an extent, we can't tease apart the differences of light availability and to an extent water, though we expected water to have less of an impact on investment in carnivory. It's an area that isn't particularly well studied in terms of how water availability impacts on carnivorous traits. It's something I've tried to do in the lab um, with a sort of pilot project with one of my undergraduates, and it's been quite difficult to get right, difficult to tweak. So yeah, good question. Something to look into.
Swanyi adds, very interesting topic indeed. Thank you. David also adds, thank you. Very interesting topic. And I think the last question is from Richard. Are you optimistic with Drosera's ability and resiliency in terms of climate change as rain and temperatures become more unpredictable or extreme? And then your like last slide, you basically said it's very hard to predict all of that. But maybe you could just talk about, are we concerned? So that's one question. But the second question is, are we concerned about like insect populations or carnivorous plants prey populations? Because they're probably not going to do well either with extreme changes in weather patterns.
Yeah, I would say I wouldn't be so yes, I'm optimistic with Drossera's ability, because and in particular my study species with Drossera rotundifolia, because they are so widespread and they occupy a range of different environments. So I would expect them to be able to tolerate those changes moving forwards. I would be more concerned with the more specialized species, those that are highly endemic or those that are restricted to specific microhabitats within these sites. Those are the ones that you need to sort of be more worried about in terms of conservation action and worrying about what the implications with those species are.
Is your Drosera rotundifolia, is it a short-term perennial or a long-term
Um,
perennial?
so generally it takes at least 2 years to grow to maturity. And then they can last for several years after that. They're relatively slow growing, as with most carnivorous plants. They die back into a hibernaculum over winter and then produce their leaves. In the UK and Europe, they emerge sort of late April, May sort of time.
I visited a Drosera population on this cliff in Japan, and the tour guide was saying that the entire population goes away in the dry season. And the only reason why they come back every year is from seed. So that's a
Oh
real
wow.
short term.
Wow, yeah.
It's basically an annual, I suppose.
Hmm.
But if those seeds don't have good weather or climate, then they're done for. Or unless the seeds are just hanging on until the right, rain occurs.
Seed persistence is an interesting one in those environments. I wonder how much turnover of seeds goes on, year to year, or whether there's a healthy bank of seeds that can last quite a long
Mhm.
time there. Interesting.
All right. Well, thank you everyone who attended this live. We had a lot. I don't know. I don't want to say we had a lot more than I was anticipating, but we did have a good, healthy turnout. And thank you so much, Chris, for an excellent presentation. This will be posted on YouTube in the next day or two. And then everyone else, you can check out our website or our Facebook page to see future ICPS webinars. And then, of course, our big day is the first Wednesday of May for World Carnivorous Plant Day. So thank you, Chris, for an excellent presentation.
Thank you very much for inviting me, and thanks all for coming along to listen to my talk.
It's not a surprise that gardeners, educators, and scientists are fascinated by these unique plants. The International Carnivorous Plant Society, or ICPS, not only love these plants, but welcomes growers just getting started all the way through professional scientists. The ICPS even started an annual World Carnivorous Plant Day to celebrate them. The free online event is held the first Wednesday of May. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.